Everyone, I'm Mr. Lukman Bolajibelo. I'll be taking you through this interesting course, Biochemistry, General and Medical 2. The course code is BCH 202. This is the course outline, cell theory. We're going to be looking at the cell theory, structures and functions of major cell components, cell types, constancy and diversity, cell organelles of prokaryotes and eukaryotes, chemical composition of cells, centrifugation and method of cell fractionation, structure, function, and fractionation of extracellular organelles, water. We're going to be looking at water, total body water and its distribution. We're going to be looking at regulation of water and electrolyte balance, disorder of water and electrolyte balance. We're going to be looking at acidity and alkalinity. We're going to be looking at pH and pK values and their effects. Now, punctuality is important, and it is very essential for you to always get your materials as and when due. Please, if you have any questions, always feel free to ask. Drop your questions on the group, or you can send your questions to me via email. The cell theory. What is the cell theory? The cell theory is just a summary of the discoveries that constitute the explanations about how the cell functions, the structure of the cells and the function of the cells. So all living things are made of cells. This is number one. This is the first of the cell theory. This is the first point on the cell theory. All living things are made of cells. Cells are the basic unit of life in living things. All new cells are created from pre-existing ones. Now, the cell, as science continue to evolve with new discoveries, we find out that certain things that were previously known to be fact, that to be theories that were postulated, were actually changed with advancement. The same applied to the cell theory. Certain modifications were made to the cell theory, giving rise to the modern cell theory. Now, the modern cell theory contains 4 statements in addition to the original cell theory. The first of these 4 is the cell contains hereditary information, that is DNA, which is passed on from cell to cell during cell division. All cells are basically the same in chemical composition and metabolic activities. All basic chemical and physiological functions are carried out inside the cell. Then cell activity depends on the activities of subcellular structures within the cell. Now, how are cells formed? Cells are formed from small compounds. In fact, it starts from the elements. You know the periodic table of elements. Those elements, some of those elements come together to form certain compounds that go on to form cells. How do they do that? The elements, there are so many elements in the body. There are so many elements found in the body. However, there are about 6 of them that are predominantly found in the body, like carbon, like hydrogen, like oxygen, and the rest. Now, these elements form compounds. A compound is a combination of 2 or more elements. These elements come together to form compounds. We have small compounds and we have large compounds. The large compounds are called the macromolecules. The large compounds that are found in the body are called macromolecules. These macromolecules assemble together to form supramolecular assemblies. An example of supramolecular assembly is our membrane. Then supramolecular assemblies also come together to form organelles. That is the subcellular organelles, like the mitochondria, nucleus, and other subcellular organelles, other organelles that are found within the cell. Now, organelles come together to form the cell. Then when cells amalgamate, when cells come together, they form what? Tissues. Tissues coming together form organs. Organs coming together form systems, and systems put together form the whole organism. So you can see that cells are central to all of these we've mentioned. So the cells are the structural and functional units of all living organisms. The reactions that take place in the body actually usually take place in the cells. So the cells are very, very important. They are considered the basic units of biological activities, and the body is composed of many, many cells. We have trillions of cells in our body, and these cells may be divided into two categories. We have prokaryotes and we have eukaryotic cells. The prokaryotic cells are the primitive cells. They are the primitive cells. They are the cells of the lower organisms, while the eukaryotic cells are the more complex cells, the cells of higher organisms like humans. Prokaryotes lack a well-defined nucleus and possess relatively simple structure, e.g. bacteria. So we find prokaryotic cells in bacteria. While eukaryotic cells are well-defined, they have well-defined nucleus and they are more complex in their structure and function. For example, the cells you find in animals and plants. This is an illustration, a diagram of plant cells and animal cells. You can see that some of the organelles you find in animal cells are also found in plant cells, whereas there are some organelles you find in plant cells that are not in animal cells. This is a table showing the organelles and their major functions. The nucleus contains DNA, and it is regarded as the brain of the cell. Why? Because the genetic information is contained in DNA, and this DNA is housed in the nucleus. The nucleus is the inner part of the nucleus that begins the assembly of ribosomes. Chloroplasts. What do chloroplasts do? Chloroplasts that are found in plant cells. They capture energy from light for photosynthesis. So plants can't make their food without chloroplasts. The other organelles are there for you, and you can see their functions. Like I said earlier, this is just an introduction. As we go deeper into the course, we're going to get more details about this. Cell fractionation. Cell fractionation is the process of separating cellular components while preserving the individual functions of each component. Why do we need to separate cellular components? It is important to do this to study the cellular components for research purposes. And this cell fractionation can be divided into three stages. We have homogenization, filtration, and purification. The homogenization is the actual breaking up of the cell. You have to break up the cell to separate the cellular organelles. So how do you break up the cell without losing the functions of the individual components within the cell? This is what homogenization is, and we have different materials, different tools we use for homogenization. We have what we call the homogenizer, and there are other things that can be used, like even the pestle and mortar. Then after that, we filtrate. Filtration is done to separate out any large debris that may be found within the homogenate you have, or tissue, or even cells that or tissue debris that were not broken up during the process of homogenization. Then after filtration, move to purification. This purification is done by ultracentrifugation, high-speed centrifugation. It is also called differential centrifugation. Why? Because you centrifuge you start from a lower speed at a less time, and then some of the cell components, some of the organelles will separate out. Then you now go for a higher speed at more time, that is at a longer duration. When you do this, more of the organelles will then separate out. Now these organelles will separate out based on their weight, based on their size and weight. The nucleus, for example, is the largest subcellular organelle. It is expected that the nucleus will separate out first before the lighter organelle. So as you move on in the centrifugation process, as you increase the speed, the lighter organelles will also separate out, and this way you'll be able to separate all the organelles. Water is undoubtedly the most important and most abundant substance in living systems. You know, we can't even exist without water. We can't survive without water. Water is the major constraint of the body, accounting for about 55 to 70% of the body weight of men and 45 to 60% of the body weight of women. So a normal man weighing 70 kg contains about 42 liters of water. This is wonderful, 42 liters of water, which is distributed intracellularly and extracellularly. Water plays a lot of roles in the body. The body is regulated by through water as well. So water plays a role in the regulation of the body, like the body temperature is also regulated through water, or water serves as a means of regulation of the body temperature and other processes within the body. There are abnormalities associated with water balance of the body. There should be a balance. You don't have more than you're expected to have. You don't have too much water, and you don't have less water than is required. So when you have less water than is required, when you lose more water than you are taking in, it leads to dehydration, and then one can also be overhydrated. That is what you call overhydration. Acidity and alkalinity. When weak acids are dissolved in water, they contribute hydrogen ion by ionizing, while weak bases consume hydrogen ion by becoming protonated. Now, the total energy hydrogen ion concentration from all sources is experimentally measurable and is expressed as the pH of the solution. So when you have more hydrogen ion in the solution, the pH is going to be acidic. When you have less, then it is tending towards the alkaline region of the pH scale. So the pH usually runs from 0. pH scale runs from 0 to 14. 7 is neutral. Below 7, 6 to 0 is acidic, while above 7, 8 to 14 is alkaline. So the farther away from 7 on the left, the more acidic it is. So a substance of pH 1 is more acidic than a substance of pH 6. The farther away from neutral, from 7 on the right, the more alkaline the substance is. So the substance with pH value of 14 is more alkaline than or is more basic than a substance of pH 8. Like I said, this is just an introduction. As we go further in the course, we're going to get more details. Thank you for your time. Feel free to ask questions. You can drop your questions on the group, or you can send them directly to me via email. Thank you very much.